Taking a look at this image of nonspecific immunity and specific immunity and the three lines of defense, we can move on to start talking about specific immunity and the third line of defense. Specific immunity allows for protection against foreign substances, but provides protection against one specific substance and not others. Meaning, as we were mentioning before, one T cell might be designed or built to attack one type of bacteria, and another T cell can attack a different type of bacteria. In order to understand this process, we need to understand what an antigen is, which is a substance that stimulates the formation of antibodies from a B cell, or causes a T cell to react in order to attack it. Antigens are found on the surface of many substances, including pathogens. We also have antigens on our own cells. However, before we're born, the lymphocytes somehow get to learn to recognize self versus non-self. And then these lymphocytes involved in the third line of defense actually know what to attack and what is self. The two types, as we've already been mentioning, are the T cells and the B cells. So let's compare these two types of cells, T cells and the B cells. T cells are produced in bone marrow, like all blood cells. However, they mature in the thymus. The thymus begins with the letter T, and T cells get their name because they are maturing in the thymus. So T cells, the T stands for thymus. These cells are involved in cell-mediated immunity. The T cells attack antigens directly through cell-to-cell contact, and we'll look at that in just a moment here. B cells are also produced in bone marrow like all blood cells. However, they also mature in the bone marrow, and bone starts with B, hence the term B cells versus T cells. They're different from T cells, though, because they are involved in humoral or antibody-mediated immunity. B cells ultimately make antibodies, which is where you get the term antibody-mediated immunity from. Sometimes people use humoral-mediated immunity, and what that refers to is that antibodies are produced by B cells to attack pathogens and are released out into the fluids of the body. The antibodies then move around the body in the cardiovascular system and the lymphatic system and attack the pathogens. Humor, in anatomy and physiological speak, doesn't mean sense of humor, but refers to fluids. And so another term that's often used is humoral-mediated immunity, because we find the antibodies within the fluids of the body. So how do T cells and B cells actually function? Let's compare them by starting with T cells. One type of cell that is involved in phagocytosis is the macrophage. Breaking that term down, macro means large, and phage means eating. And so a macrophage is a large phagocyte that will go around and ingest antigens or pathogens within the body. As is being indicated here, it is phagocytizing a bacteria. The macrophage then breaks down that pathogen, and it pushes the antigens to the membrane surface of the macrophage, a process called antigen presentation. It's presenting the antigens on the surface so that it can help activate the appropriate T cell that is designed to attack those antigens. So as we see here, there are receptors on the T cell that when they match the appropriate antigen being presented, they will undergo the process of division. They will produce a number of clones. So the T cell is activated, and the first thing it does is it divides. And then it divides again. And then all those cells divide again and again, so that many of them are produced. And there's four major categories of the types of T cells that are produced. The first is the killer T cell. This is the cell that actually goes and kills the antigens around the body through cell-to-cell combat. As we said, T cells are involved in cell-mediated immunity. Helper T cells go and stimulate other T cells and B cells that are appropriate for getting involved in attacking the antigens and pathogens that are within the body. Suppressor T cells inhibit T cells and B cells. So the suppressor T cells are involved when the attack is over. When the pathogen has been successfully removed from the body, the suppressor T cells go around to the T cells and B cells involved in that immune reaction and tell them to stop attacking. A final type of T cell that's important is the memory T cell. These cells, when produced from the T cell activation and the clone division, are memory T cells that then remember the antigen for future encounters. They're actually not involved in this initial response by attacking the pathogens in the body. They sit around the body and wait for the next time that you get the infection via that same pathogen. This is important in terms of our bodies being able to fight off a second infection from pathogens. We'll take a closer look at that and its role in vaccines in just a few moments. So for B cells, the first process is very similar. Macrophages phagocytize the antigens or the pathogens, and they present those antigens on their surface. Then we have the activation of B cells and helper T cells. And this is important, as we were just mentioning, helper T cells help to stimulate T cells and B cells. What they will do is they will go to B cells as the macrophages present the antigens, and they will release a chemical called lymphokine. If that B cell is the appropriate B cell to mount a response against the pathogen, the lymphokine will help speed up the cell division of that B cell. Again, there'll be the formation of clones. That B cell will divide, and then those two cells will divide. Then those 4 cells will divide, and we'll form plasma cells, many of them, that then secrete antibodies, and memory B cells. Just like the memory T cells, these remember the antigen for future encounters. They do not produce antibodies for the time being. So those plasma cells secrete antibodies, but we want to understand the antigen-antibody reaction. So let's take a closer look at that plasma cell. It secretes antibodies which have this chemical structure. It looks like a Y. On the tip of the Y, where the yellow and the blue meet, there is protein that is coated in such a way that it is created to attack a certain antigen. Here's where the specificity is important. Maybe this antibody is built in order to attack this kind of bacteria. So the tips of the antibody here will bind to the chemicals on that bacterial cell, and it will act as a marker to allow the complement chemicals that we looked at earlier to come in and attack the bacteria, punch holes in the surface. It will also attract phagocytes to come in and phagocytize the bacteria. But the key here, as we mentioned earlier, is this is specific. This plasma cell that we saw here produced antibodies against this type of bacteria. But we have many different types of plasma cells. Each of them are going to produce slightly different antibodies in terms of the chemical protein sequence at the tip of those antibodies. If we get another type of bacteria shown here into our body, these blue and yellow antibodies would do nothing to attack these bacteria. But another plasma cell might get stimulated by a macrophage to produce its antibodies, then will go and attack that bacteria. Because again, the protein sequence or region at the tip of the antibody is tailored to attach to those chemicals on this different bacteria. So let's differentiate now between naturally and artificially acquired immunity. In immunity, we have a few different categories that we can break it down into. The first is genetic or inborn immunity. What we're referring to for genetic or inborn are just differences between genetics among people. To use an extreme example, there are certain diseases that, let's say, your pet might get, but that you would never get. Your dog might get heartworm, but heartworm is not something that humans can contract from their dog because our genetics work in such a way that that worm would not be able to survive inside our body. Similarly, there's differences in genetics among people, where some people are more prone to certain diseases than other people. Acquired immunity actually breaks down into 2 categories: naturally acquired and artificially acquired. The difference between the two is in naturally acquired immunity, someone actually gets the disease. Artificially acquired immunity is more man-made, if you will, and we'll take a closer look at that one in just a moment. But each of these break down into two subcategories, active and passive immunity. So we have active naturally acquired immunity and passive naturally acquired immunity. The difference here is that in naturally acquired active immunity, you actually get the disease. You mount a response to the disease with phagocytes attacking the disease, and then your T cells and B cells mounting a response to the disease as well. Ultimately, you end up producing memory T and memory B cells in order to actively fight off the disease if you were to acquire it again. Passive naturally acquired immunity is passive because it's from mom. The example of this is that when a mother gets infected with a disease, chances are her offspring is also getting infected with the disease. The mother's immune system mounts a response. Her phagocytes attack the disease, her T cells and B cells also mount a response, specifically with her B cells producing antibodies against the disease. The antibodies that she produces actually will enter into her milk that she will breastfeed to her offspring. The offspring, who may get the disease, doesn't have an immune system ready to fight off that disease, but it gets antibodies from the mother. So this is naturally acquired, but it's not the individual, the baby, actually producing the response, and therefore it is a passive, naturally acquired immunity. Artificially acquired then has active and passive artificially acquired immunity. Active artificially acquired immunity involves vaccines. And we'll look at that in more detail in just a moment. But a vaccine is scientists have taken a disease, gone into a lab, killed off the disease, but then injected patients with the dead bacteria or the dead virus. The chemicals or the antigens that make up the bacteria and virus are injected into the person's body, and that person's body actually mounts an attack against that disease, even though they didn't naturally get the disease and are not at risk for developing the disease. Passive artificially acquired immunity is a situation where perhaps somebody else has got the disease, they have mounted immune reaction to it, and they've produced antibodies that are now within their body fighting off that disease. A second person who cannot fight off the disease for some reason can actually be given antibodies from the first patient and have them injected into their body to fight off the disease. So it's somebody else's antibodies that are actually in the second patient's body fighting off the disease. And there will be no memory cells in this passive artificially acquired structure. Again, the difference here between the active and passive is whose body's mounting the response, as well as, just like in the naturally acquired, there's a difference in terms of memory cells. So when given a vaccine, your body will produce memory T and memory B cells that will then remember the disease for the next time when you might actually contract the real live disease. For passive artificially acquired, the antibodies will fight off the disease in your body, but you will have no memory cells for the next time you get ill. You would actually need antibodies yet again to fight off the disease. So what is a vaccine ultimately? We've mentioned it already, but in the lab, we take some bacteria and we can actually break down the bacteria such that we have all the antigens that make it up, but the bacteria is no longer surviving. We take those bacteria and we place them into a syringe, into a needle. A patient comes along and we give them the vaccine with those dead bacteria or dead viruses within the vaccine. The person's body then mounts an immune response against those chemicals. And then when they actually get the disease in the future, they will mount a larger response, and we'll take a look at that here. So if we look at the antibody concentration in serum, we have a primary response indicated here after the first exposure to a disease. So we have first exposure shown here on day 0, and the primary response is such that we produce a low level of antibody concentration in the serum, and it takes upwards of 2 months in order to be able to fight off the disease completely. However, when we create this primary response via a vaccine and the patient is not going to get sick, it allows for the secondary response. On secondary exposure, when actually contracting the disease, your body will mount a much greater and faster response, such that after only 30 days, you have much more antibody concentration in the serum than after the initial exposure. This has been very effective in fighting off diseases that were once very common and very debilitating in the world, because after exposure to vaccines, our bodies have been able to fight them off much faster and with less negative repercussions.